audience.
work capacity of the UK in sandfly and leishmania interactions?
Probably more than that.
Well,
Practical expertise.
Matt, let me ask you something. Usually we record the sessions.
Yeah, that's fine.
If you are okay with that, we how can I say, broadcast them in YouTube. But if you are not okay with that for unpublished results or something like that, it's fine. We just keep it.
Let's record
private.
it and then let me think about it. But I'm sure I'll be fine with it. But I just need a little.
You can decide that later. I'm just warning you because you will realize the symbol of recording in your screen.
Yeah. I've just
Don't be scared. We will not use it for nothing unless you allow us.
That's fine.
Okay.
Okay. But I think what is interesting, if you have a specific part of the presentation that you do not want it to be published,
we can remove.
online, the Itabajara can edit it, and that's okay.
Yeah.
So
Professor Itabajara is very good doing
let
that.
us know.
Yeah.
He can extract seconds of your presentation if you want.
Extract and add specific things.
We can put words in your mouth. It will be
we can change words. Put pictures.
Tabajara, this is very popular now in Brazil. People doing this everywhere.
Exactly.
Yes.
We are working in
fake news, fake research.
No, it's a joke.
So let's wait. Logulo, when we shall start?
Yeah,
it's time already, or
yes, you can.
Okay.
No problem.
Rod, can you please introduce Matt? I thank you so much for accepting this invitation. And I'm very pleased of having you, Matt, presenting, and Rod introducing Professor Matt Rogers. Thank you so much.
Okay, so yes, it's a real privilege to introduce Matt Rogers to you lovely people, mainly based in Brazil. But I've known Matt for, I don't know, 17 years or more. I can't work it out now, but just a little potted history about Matt. I think Matt graduated with a biology degree at Imperial College in the 90s, 94, I think. And then he sort of moved up north to Liverpool, where he gained a master's in parasitology and entomology. And that's when he started working on sandflies and leishmania. And he did, after that, he started to do a PhD on leishmania sample interactions and started a long collaboration with Paul Bates and went on to do a couple of postdocs, I think, involved in the transmission of leishmania through sandflies. And that's where I appeared and met Matt. We sort of started to work together in Liverpool. And then Matt went back down south to London and returned to Imperial College, where he started working on immunology of leishmania infection in sandfly bites in Ingrid Muller's lab at Imperial, and then various permutations. He got awarded several other fellowships, and he moved to London School of Hygiene Tropical Medicine, where he is now. And he's now Associate Professor of Vector Biology in the Department of Disease Control. He's got his own sort of research program, plus manages to do a bit of teaching on master's courses and various diploma courses and everything. So yeah, what can I say? On a personal note, I think sort of Matt is an amazing, very precise and creative scientist. We always have fantastic sort of conversations about research and the science. Matt's published about 50 odd papers, including Nature paper, PLOS pathogens. So it's published very well, very widely. Got amazing technical skills in the lab as well, not just good at writing papers and grants, but he can do anything you want to do with a sandfly, or sandfly gut. Matt can do it. And if he wants to get a sandfly to feed on something, Matt can do it. So he's the go-to guy if you want to do something with a sandfly. So, and I think the past year or so, Matt's shown his dedication to the cause. I think he's kept his colony in his kitchen at home with his 2 kids and his wife. And I'm not sure how much his wife approves of the situation, having a Jacobina colony in his kitchen, but I think that's what's happened. I don't think the kids have been drawn into the blood feeding yet, but
Once or twice.
that might be on the cards in the future. So yeah, Matt is coming to give us a talk about, I think, new approaches and new insights to Leishmania transmission. So we look forward to hearing more about it from you, Matt. Take the floor, please.
Thanks, Rod. That really is a wonderful introduction. Thank you very much for being a good friend as well. So yes. And thank you everybody for the introduction and the opportunity to speak to you about the topic that I love and I passionately, I enjoy working with every day of my life. And that's the kind of unique biology behind Leishmania sandfly interactions, and particularly focusing in on Leishmania transmission, which I'm particularly interested in, and that's where my career has kind of gone. But to kind of just preach to the converted, this is to bring people up to speed with leishmaniasis. I now realize that that's probably not entirely unnecessary, but we have leishmaniasis, which is a debilitating protozoal infection affecting very poor communities around the world. And depending on the species you have, it can manifest as a variety of different pathologies, from simple cutaneous leishmaniasis, mucocutaneous leishmaniasis, which is a bit more complicated and certainly more destructive, and then visceral leishmaniasis, which is life-threatening if not diagnosed and treated early enough, and can affect a potentially huge population of people in a wide number of countries, with an annual incidence of about 2 million cases, resulting in about 500,000 or 400,000 deaths per year. The burden of cutaneous leishmaniasis and the social aspects and stigmatization associated with infection is much bigger and very, very hard to put a finger on and put a number on. However, this just shows you the very wide geographical range of leishmaniasis over the world. You guys are in the hotspot of foci, one of the major foci for visceral leishmaniasis and mucocutaneous leishmaniasis. But it does stretch across the world, in Mediterranean and North Africa, right through Africa into the Middle East and beyond. Oh, sorry, a bit slow here. And this is the Leishmania life cycle, which cycles between a female sandfly, blood-feeding female sandfly, and a mammalian or vertebrate host. And so the female sandfly takes up mastigotes with their blood meal, and then those parasites quickly transform into a flagellated promastigote form, and we get a sequence of development which ultimately results in infectious forms known as metacyclic promastigotes, which are then deposited by the sandfly bite when she refeeds on another host. And those parasites are then phagocytosed by macrophages principally, where they then complete a final transformation into an amastigote form, and the cycle then continues. And it's been my kind of early focus of my career to look at the interaction between the sandfly and the parasites, and to quantitate the development of the parasites inside sandflies in a bit more detail. And this slide is one of my favorite slides, and I think it sums up what I'm all about and what I'm particularly interested in. This is a very rare image captured by Henry Short in 1943 of, unfortunately, it's not an infected sandfly, but it's a sandfly caught in the act of blood feeding on mouse skin. And it shows you some lovely detail, not least the pool of blood that it's feeding on underneath the skin, and hence sandflies belong to that limited range of vectors which are pool feeders and lacerate the upper dermal capillary loops to obtain their blood. However, so sandfly interactions is one of my main focuses of study, and I believe that if you understand that and the transmission mechanism in detail, and of course on how those two things impact on the host parasite vector interaction in terms of immunity, and also you need to consider the behavior of the infected vector. That will give you a much more global idea about the interactions which are critical for a successful transmission. And once you know those things, you can really then inform the kind of the major topics of leishmaniasis, not least the immunology of leishmaniasis, which would then, once you get a better picture of that in terms of natural infection, you can then feed that into better drug and vaccine screening programs. And ultimately, you can incorporate this knowledge to improve your models of epidemiology of leishmaniasis. So, this slide just illustrates to you the kind of diversity of developmental programs that Leishmania undertake inside their sandfly vectors. And as you can see, there are three main ways in which the parasites can develop within the sandfly. Today's lecture is going to focus on the model the development that I mostly work with, which is Leishmania and Leishmania, the subgenera Leishmania. And they undergo a suprapilarian development. That means they don't go into the hindgut, they leave the peritrophic matrix encased blood meal and migrate anteriorly towards the stomatal valve. However, there are another major subgenera, Leishmania-viana, which includes a period of hindgut development before they migrate anteriorly towards the mouth parts. And that comprises the New World species of Leishmania. And then you have the outlier, which is Sora Leishmania, lizard Leishmania, which have a hypopilarian form of development. And they don't go into the anterior midgut at all. They remain almost entirely inside the hindgut. And it's assumed that their transmission is by ingestion of the infected vectors. And so I began by, as I mentioned, looking at the different morphological forms of Leishmania with pool baits in sandflies, using Leishmania mexicana, an experimental model with Lutzemeyer longipalpus. Not least because we had the flies and we had the parasites, and we can grow the parasites very well. And a lot of genetic manipulation back then was being done with Leishmania mexicana. So it was a good model to start with. And the graph shows you very nicely how the different morphological forms wax and wane throughout the infection in the sandfly. And you can see a very nice succession very early on in the developmental sequence of the procyclic promastigotes, nectomonad promastigotes, leptomonad promastigotes, and then the all-important metacyclic promastigotes, and although a minor population, haptomonad promastigotes. And in the next few slides, I want to kind of just introduce you to these sort of major players in the development of Leishmania within the sandfly. It begins with the amastigote, which is taken up with the blood meal, and they get deposited in the abdominal midgut, where the blood meal is encased in a peritrophic matrix. And these are relatively small. They don't have any visible by light microscopy flagellum, although they have a kind of peg-like structure, which very minimally pokes outside the flagellar pocket. And their kinetoplast is parallel to their nucleus, and they are immotile. But their role is to start the infection in the sandfly. And then within 24 hours, depending on the species, the amastigotes transform into the first procyclic promastigote, which are the first promastigotes, which are called procyclic promastigotes. These are slightly enlarged from the amastigote form. They push out a small flagellum, which is weakly motile. And as a result, they have sluggish movement. And they tend to form nests or nidai within the early blood meal phase of development. And they're not around for that long. Well, they don't peak for very long inside the sandfly midgut. But what they give rise to is what if you have favorites, this is my favorite, the nectomonad promastigote, which is probably the most dramatic of all the cell forms inside the sandfly. Nectomonads are extremely elongate in their cell body. They have a long flagellum and they are highly motile. And that's their main job. Their job is to colonize the fly. And so, as I mentioned, they're very long, and their job is to escape the peritrophic matrix, encased blood meal, before it's at the end of digestion, and to resist defecation. And these forms don't replicate, but their job is to colonize the rest of the anterior midgut. And so if we look carefully by dissecting the gut into different parts over the span of the infection, we found that nectomonad promastigotes are the first to arrive at the stomatal valve. So their job really is, because of their strong swimming capability, to get to that anterior station and to establish the infection for onward transmission. Then we have a cell form which we kind of we identified, and that's the leptomonad promastigote. Leptomonad promastigotes are the product of a transformation of the nectomonad promastigotes, and after they form this leptomonad promastigote, which is a more of a contracted form of promastigote, sometimes called a short nectomonad, they then reinitiate division as these forms, and these forms accumulate in the anterior midgut, and their numbers, their presence is very closely associated, tied in with the accumulation of a gel-like material that we termed Promastigote Secretory Gel. We'll come on to this later, but the job of the leptomonads then is to condition the gut by secreting this gel to adapt it for successful transmission. And these forms, the leptomonad promastigote forms, we believe are the precursor of the metacyclic, the promastigote, the infectious form. But before we get to the metacyclic promastigote form, we have another cell form to consider, and that's the Haptomonad promastigote. And these are very unusual and very specific cell forms. They have a modified flagellum, which attaches to the chitinous surfaces within the sandfly midgut, principally the stomatal valve, although it's suspected that Viania species also produce Haptomonad promastigotes in the rectum and in the hindgut. However, the Haptomonad promastigotes are sometimes considered altruistic forms, so it's assumed that they're not necessarily transmitted because they're attached, but what they do is probably initiate the biological plug of parasites at that anterior station, the stomatal valve, and start the kind of deformation of that important structure which separates the midgut from the foregut and allows parasites to access a position for transmission. And as a result, when you look at them under the electron microscope, they can sometimes appear dead. They're electron-lucent. However, I think they have an unappreciated role in Leishmania transmission. And then finally, we have the stars of the story, I suppose, which are metacyclic promastigotes. These forms are, again, dramatically different to a lot of other promastigote stages in the sense that they're very thin cell bodies. They usually have a small cell body length and a relatively long flagellum in relation to their body. As a result, they are very active and they're very good at moving within that limited space within the anterior midgut. And they have a number of pre-adaptations for successful infection of the vertebrate host after they're transmitted. And so they very significantly modify their glycocalyx of LPG, and they extend that molecule, they double or triple its size, which lends itself to resisting the harsh environment within the mammalian or vertebrate host. It can protect itself from the membrane attack complex, which is the end result of the complement cascade, and indeed utilize components of the complement cascade to access receptors on their host cell, the macrophage. And by doing so, they gain immune silent entry into those cells by dampening down an early IL-12, an important instructional pro-inflammatory cytokine released by mastigotes when they're infected. So they can suppress the immune system from the very start of the infection. They also upregulate a glycoprotein which has protease activity, GP63, which also can protect them from complement. And by modifying their LPG and certain species modify the sugars which branch off the LPG, they can ensure that they don't reattach to the sandfly midgut, which ensures that they're in the lumen of the sandfly midgut and available for transmission. And it's something we noticed that we'll come back to in the talk, which is the just a simple observation that within the gel, the metacyclics, compared to all the other motile non-metacyclic forms, can move very freely within the gel, whereas the other promastigote forms really struggle. And that's something that I think is critical for successful transmission. And as I mentioned, we'll come on to that in a bit later. And the graph on the right shows the infection profile of Luctomyia longipalpis with either Leishmania mexicana in the squares or Leishmania infantum, Shigazi, in the circles. and just shows you the accumulation of metacyclogenesis can be very different between species, even in the same vector. So it's something that I've certainly tried to pay attention to through my research. Okay, and add that all up and you get this nice life cycle, first published by Paul Bates, and just shows you quite nicely that you have two main replicative cycles early in the blood meal for the procyclic promastigotes, and later on in the anterior midgut with the Haptomonad promastigotes, which is sandwiched, which in between them you have the non-replicative Haptomonad promastigotes, which have the job of migrating to the anterior midgut. And you also have the Haptomonads, which attach to the stomatal valve and manipulate the sandfly vector. And then the Haptomonad promastigotes generating metacyclic promastigotes for transmission. Okay, this is in a little bit more detail, but just shows you in a sort of cartoon form all those sort of players spatially within the sandfly midgut, going forwards towards the stomatal valve. I won't linger on that, but just suffice to say that, you know, I'm particularly interested in, well, I suppose most of this, to be honest. I'm particularly interested in transmission at the anterior midgut, and how the parasites adapt their sandfly vector for transmission, and also how they initially colonize that vector by attaching to the midgut and resisting the defecation process. So this is a very nice slide I've stolen from Tiago Serafim, and it just shows you transmission, what we know currently about the different elements of transmission, not necessarily how they interact, but their presence. And so we know that metacyclics are very important for infection. They're the infectious, the principal infectious form, and they're deposited into the skin during an infectious bite. But there are a number of other vector-derived components which have been proven to have a very significant effect on influence on that infection. And not least, we have the vector saliva, which is a whole pharmacopoeia of molecules which manipulate the blood feeding process, but also participate, have been co-opted for Leishmania infection. You have the bacteria, which are commensal to the sandfly midgut, which are shown to influence infection. And also the bacteria from the host skin, which has been pushed into the blood, sorry, the transmission site. You have, and then you have exosomes, which are packaged and processed and then released by the parasites inside the sandfly midgut. And we have this gel, PSG, Promastigote Secretory Gel, which is a proteophosphoglycan-rich matrix, which is secreted by the parasites, the Haptomonad promastigotes inside the sandfly midgut, and which is co-inoculated with the parasites during a sandfly bite, an infectious bite, and encourages the regurgitation of parasites and gel into the skin. It really is an important molecule which adapts the sandfly for efficient transmission. So just to pull back a bit, what do we know about transmission and know about infectious bites? Well, unfortunately, we still don't know too much about the transmission event itself. And we know certain key things. We know that the number of ingested or regurgitated parasites range from very low numbers up to quite high numbers per bite. And we know that transmission is more likely to occur when a gut is enriched for metacyclics. And we know that the parasite dose correlates very reasonably with the midgut infection intensity, and related to that, it also correlates with the amount of gel that the sandfly is carrying within its sandfly midgut. And then finally, we have this last fact which I'm particularly interested in, that the composition of the infective dose is highly enriched for metacyclic promastigotes. And that flies in the face of what is actually present in the sandfly during transmission. It's actually quite a mixed bag of promastigote forms even during a mature infection in a sandfly. And so the metacyclics will only really constitute, well, certainly for Mexicana, during a primary infection, around about 50% of the promastigote forms, and the rest is usually made up with leptomonads, a smaller population of nectomonads, and then you've got the haptomonads, which are attached to the valve. And so how do the parasites achieve that highly enriched metacyclic dose? And so we began to explore that a little bit closer, and we delved into the realms of qPCR, quantitative PCR, to see if we could quantitate the number of parasites transmitted by individual sandflies, and hopefully maybe even tease apart what proportion of those parasites were in fact metacyclic. And so on the left, you have the data from individual sandfly transmissions in terms of total numbers of parasites transmitted using an ssRNA quantitative PCR system, which has been used before by Kimblin and others. And we see, like them, that you get this kind of bimodal distribution. It's quite heterogeneous, but there appears to be a bimodal distribution of the numbers of parasites transmitted, where the majority deliver relatively low numbers, 10 to 100, maybe 400 or 500. And then you get another population which can potentially transmit a larger number of parasites. So not every infectious fly is equivalent in terms of the number of parasites transmitted. And when we looked for metacyclic markers, and we screened quite a few, and we came upon SHRP as a good marker for differentiating infectious and non-infectious promastigotes, metacyclics versus all the other promastigotes. To our surprise, we saw that when we analyzed these individual transmissions, we saw that although the majority, when you average them in terms of the percentage of metacyclics that were delivered for each fly, when you average them, they all come out at a pretty high percentage of metacyclics. But when you drill down into the quality of each transmission according to the amount of parasites they transmitted, you can see that this is very heterogeneous. And as the dose increases, as the number of parasites being transmitted increases, you can see that there are an increasing proportion of non-metacyclics being transmitted by those flies. And so at the high end of transmission, high dose transmission, you tend to get more non-metacyclics creeping into that dose. And we were fascinated to really kind of explore this a bit more and certainly see what the consequences of that was on infection. And as I mentioned, we played with this a little bit more, and I find this these graphs quite fascinating. And what they show you is that the graph on the left shows the relationship between the numbers of parasites transmitted and the load of parasites in that sandfly, and also the quality of that those transmissions. And you can see it's roughly linear. The number of parasites transmitted is roughly linear to the load inside the sandfly, as you'd expect, until you get very high infection levels in sandflies. And then the number of parasites being transmitted drops. But when you look at the quality, you can see that the quality of those transmissions is either best when you've got low numbers of parasites inside the fly, or at the other extreme, very high numbers of parasites inside the fly. And that's reflected in the graph on the right, where you can see the dose quality in a bit more detail. And it really plummets when flies have a relatively high dose at the higher end of the dose level, but then it rebounds and becomes more metacyclic enriched when you get very high infection levels. What I'm particularly interested in is the other extremes where you've got low numbers of parasites. And this is probably more reflective of what's happening in nature anyway, because it's been shown that sandflies pick up relatively few parasites, certainly fewer than we have anticipated in our experimental models. And so this may actually reflect a more true situation where you have low-level infections, which, as a result, can transmit high-quality infectious doses for metacyclics. And so you can sort of maybe ask certain questions when, for example, like when is transmission optimal in this experimental system? And so to do that, we looked at the quality of transmission by different ages of infection, different days post-infection, from 5, 7, and 9 days post-infection. So at 5 days, you're starting the metacyclic process. You get a few metacyclics coming through for Leishmania mexicana. And by day 7 and day 9, you've got mature infections where you've got infections with 40 to 50% metacyclics in their midgut, although day 9 would have more parasites in general and more gel. And as you can see with the bottom bar graph, which reflects the one above it, you can see that the quality, so the darker the band means the more enriched it is for metacyclics. As the infections mature, you get more non-infectious promastigotes, or more dirtier transmissions, if you like, with very advanced infections compared to the previous days. And if you bin that according to the number of parasites transmitted, then yes, you can see that for day 5 and day 7, the transmissions, although on average are lower, certainly for day 5, but by day 7, you get very reasonably high numbers transmitted and a high proportion of metacyclics being transmitted as well. So this kind of raises questions about when are sandflies optimal for transmission, which I think certainly needs a lot more exploration. But going back to that question that I raised earlier, what is the consequence of having a mixed transmission? Now that we know that the majority of sandflies transmit a high population of metacyclics, but nevertheless still can transmit non-metacyclic promastigotes, how does that impact upon the infection? And so we conducted some mouse infections in bulb-C mice using this mocked up range of transmission scenarios by injecting into foot pads either 10 to the 4 metacyclics, 100% metacyclics, 10 to the 4 nectamonads, so 0% metacyclics, a lower dose of metacyclics, but nevertheless still 100%, and then a mixed dose with 50% metacyclics and 50% nectamonads. And you can see that the 100% nectamonad infections didn't take off. They weren't any really visible lesions there, as you'd expect for a non-infectious form. And the metacyclics, 100% metacyclics, performed very well. So you've got these middle two lines here representing the 100% metacyclic doses. But the evolution of the lesions was much faster and much more aggressive in those infections which were mixed, that had 50% metacyclics and 50% nectamonads, non-infectious promastigotes. And when we looked at the parasite burden, this middle graph, in those lesions at the end of the infection period, we saw that there was fewer parasites in those larger mixed infection lesions compared to the 100% metacyclic lesions. And so you've got this scenario where you've got more parasites but less metacyclics, and you generate bigger lesions, but those lesions contain fewer parasites, fewer amastigotes within them. And then we expose sandflies to lesions from the mice in the same infectious cohort, and we can see the same translating into sandfly infections too. So mice that were infected with a mixed population of metacyclics and nectamonads are poorer at transmitting back to sandflies. So it reflects the availability of amastigotes in those lesions, even though those lesions are much bigger. So you've got something very unique going on there, which I think probably needs more work on. And certainly when we drill down into the immunology surrounding these transmissions, we saw that the mixed population infections generated more chemokines and early pro-inflammatory mediators such as IL-1 beta, which certainly are not conducive to parasite survival in the skin. And so all of that is very well and good for a primary transmission, but then we've got this fantastic paper, which is a kind of an evolution of some earlier work in the 90s by Dia Al-Naim and Richard Ward, which really explored the influence of a second blood meal on the transmissibility of leishmaniasis. And very strikingly, very dramatically, it was shown that you have augmented transmission when you take a second blood meal by increasing the population of metacyclics in your sandfly by either promoting the division of leptomonad promastigotes or by a new process of metacyclics reversing their biology and transforming into a leptomonad form, and thereby increasing numbers in the sandfly and increasing the proportion of metacyclics. And this begs the question, how does this influence the quality of transmission from these flies? Bearing in mind what we know about a primary transmission being potentially mixed, even at the best of times, in an infectious sandfly. And so that's something I'm particularly keen to look at in certainly in more detail. And so now I'll turn my attention to how the Leishmania really can successfully adapt their sandfly vector for transmission in more by the secretion of a glycoprotein-rich gel known as Promastigote Secretory Gel. And the top, the cartoon shows the typical position of a gel plug, which resides within the thoracic midgut and stretches into the anterior midgut, and certainly has the effect of opening the stomadeal valve, or contributes to that being open. In the middle panel at the top shows a single PSG plug which has been dissected from a heavily infected Leishmania mexicana infected Leishmania-sand sandfly, which shows you that we can manipulate this material very easily for experimental purposes. The immuno image shows the parasites stained in red embedded within the gel, which is stained in green. This is a cross-section across the infected thoracic midgut. And the scanning electron image shows that at much higher magnification, the gel is actually more of a fibrous matrix, which the parasites seem to be sort of trapped within, a bit like a net, really. But to cut a long story short, that the gel is particularly important for transmission and infection for a whole host of reasons, including the fact that it protects parasites from digestive enzymes early in the blood meal, when it's being secreted by the early blood meal stages. It promotes the regurgitation of parasites when it forms this gel-like blockage, the blocked fly phenotype. It can alter the mammalian host immunology in favor of the parasites. So the gel can actually skew the activation of macrophages towards an alternatively activated state, which is the best scenario for Leishmania, because those cells actively produces lots of compounds such as spermine, spermidine, which are intracellular nutrients for the parasites. But going back to the sandfly, it can certainly alter blood feeding ability of the vector very, very dramatically. And as a result, it can induce a persistence behavior. It can encourage the flies, infected flies, to refeed after it's been interrupted. And that's a key aspect of encouraging more transmission events. And so I'll go back to that observation that we made, that metacyclics appear to be more motile within the gel compared to other promastigote forms. And we did a very simple experiment to begin with, and that was to cut the freshly dissected infected PSG plug into 3 parts and analyze the proportion of different promastigotes in those different parts. And as you can see, the poles of the gel plug are more enriched for metacyclics compared to the middle of the gel. And so it seems that the metacyclics are freely able to kind of move to the kind of periphery of the gel. And to complement that, we did a similar experiment, but instead of dissecting the gel, we punctured the infected sandfly gut, and we collected the emerging parasites over time, and then analyzed the proportion of metacyclics that we collected. And we can see that even for up to 10 minutes of collecting parasites emerging from a single sandfly gut, it's pretty enriched for the metacyclics in the light gray portion of each bar. And then, as I mentioned earlier, that's in direct contrast to what's actually in the fly at that time. When we analyzed the profile of infection in the remaining gut, and you have certainly more leptomonad promastigotes and nectomonad promastigotes in the gut compared to what has emerged from it, from that puncture. So it demonstrates to us that there is some differential mobility of the promastigote forms within the gel. And then we hypothesized that perhaps there is a stage-specific adhesion going on within the gel, and so it retains the non-infectious forms, but allows the infectious forms to move freely within it. And to analyze that, we quite simply did an adhesion assay using purified populations of the different major promastigote forms. And lo and behold, in the top left-hand panel, you can see that the gel can adhere the nectomonad population and the leptomonad population very, very strongly compared to the metacyclic population, and indeed the procyclic population, which should never actually be, well, relatively few should experience being within the gel itself. And then to put that in a more sort of 3D context, we performed what we call capillary migration assays, where we load up capillaries with either PBS or gel from freshly dissected infected sandflies. And we introduce known populations of parasites at one end, and we collect those which emerge at the other end over time. And these two graphs at the top here show the profile of migration of metacyclics or nectomonads, either in PBS or the gel. And the nectomonads can freely move through PBS, as you'd expect, but get stuck within the gel and they don't come through as much. In contrast, the metacyclics can move freely through the PBS or the gel equally, and they come through quite quickly and in quite high numbers in either medium. And so that sort of really sealed the fact that we probably do have a stage-specific adhesion going on inside the gel, inside the fly. And our immediate attention was drawn to LPG, bearing in mind that LPG is a very important ligand to attach to sandfly midguts. And when we repeated our attachment assay on PSG, but using wild type or LPG2 deficient parasites, which can't synthesize any phosphoglycans, LPG1 deficient parasites, which can synthesize everything except LPG, or the LPG1 add-back, so they should be like wild type. We can see that the absence of phosphoglycans or LPG really ablates their ability to attach to the gel. So LPG is certainly involved in this stage-specific attachment process. And when you put those LPG deficient parasites into this capillary migration assay, and the two small graphs here in the middle, you can see that nectomonads, unlike their wild type counterparts, they can now move freely through the gel, and the metacyclics behave much like the wild type too. So the big difference is in the nectomonads. The nectomonads that don't have LPG on them can move through the gel quickly. And so when we compared individual transmissions of these main infections, wild type, LPG deficient parasites, or the ADBAC. And we looked at the ratio of this metacyclic specific transcript as a proportion of the of ssRNA, which is a transcript which detects all Leishmania forms. We can see that those transmissions which have the least amount of metacyclics coming through are the LPG1 deficient parasites, which kind of backs up what we suspect is happening inside the vector, that the gel is retaining those non-infectious forms as best as possible to allow the metacyclics to be transmitted. Sorry, this is being slow. And so, just to take you through some of the new approaches that that we are trying to experiment with, and to see if we can use effectively to complement our approaches to study Leishmania transmission. I've reached out to a number of labs that specialize in certain biophysical or biochemical techniques that I think would be very, very sensible and very instructive to analyze Leishmania transmission or Leishmania sandfly interactions. And they're listed here. And so we have surface plasmon resonance, or BioCore, which is a much better way, a much more improved way of getting better quality data on the kinetics of binding between ligands. Microreology, which is looking at the viscosity of certain media with the aim of looking at the PSG itself. Atomic force spectroscopy, which is a fantastic new, well, not new technique, but is a fantastic biophysical technique to really get accurate data on the forces of attraction and a spatial distribution of those forces over a substrate. I'm also collaborating with people who do holography and analyze the movement of particles in a 3D environment. And then I recently participated in a project which used microcomputerized tomography to look at infected sandflies, and hooked up with a mathematical modeler who can really unlock the power of the data that we collect in our infections. And then hopefully in the future, a collaboration might be fruitful to manipulate not only Leishmania, but sandfly embryos as well, to probe very specific questions. And I've picked out two which have been particularly fruitful so far. And the first involves atomic force microscopy. And this goes back right to the beginning, where we have this need for the parasites to attach to the sandfly midgut to enable it to successfully colonize it later and be transmitted by it. And so this goes back to the attachment phase of the development. And as we're probably all aware, the LPG is very important for attachment, certainly for permissive sandfly, Leishmania sandfly combinations, of which there are relatively few out there. Leishmania major and Phlebotomus palpatasi being the best characterized so far, where you have a very specific galectin on the surface, projecting from the surface of the sandfly midgut into the lumen, which captures the galactose on the LPG projecting from the LPG of Leishmania major. But the other major group of vectors which haven't really been explored, or certainly explored in any great detail except by Peter Wolf's group, are the permissive sandfly vectors, for which there are many. Unfortunately, we grow one of them in London, which is Lutzmeier longipalpis. And so this data here just shows you the kind of scenario we have between Lutzmeier longipalpis and Leishmania mexicana. And so if we extract LPG from nectamonads or metacyclics, you can see in the middle panel here of midguts which have been opened and probed with LPG from the different promastigote forms. The top two panels are no LPG, so you've got a control there. The middle two panels represent the binding of nectamonad LPG, and the bottom two panels metacyclic LPG. And so it just shows you quite nicely that nectamonad LPG is very capable of binding to the microvilla of Lutzmeier longipalpis. And when we perform traditional midgut binding assays, we see that the LPG deficient parasites, like the metacyclics, cannot attach in any great number to the sandfly midgut. But if you add back the LPG1 gene to the nectamonads, you can restore the binding. And you can, to a certain extent, block that binding by co-incubating with an anti-LPG antibody. And the bottom panel shows some of the is a competition experiment using various sugars during that midgut binding process. And we can see that GalNAc, amongst them galactose and glucose and glucnac, can effectively interfere with that binding process. And this leads us to the observation that Miskova in Peter's lab 2016 characterized a mucin which commonly coats the permissive sandfly midgut. And there is, this mucin is particularly enriched for O-type glycosylation sugars, of which GalNAc is certainly one of them. And so we thought, let's use atomic force microscopy using GalNAc as our molecule that we wish to probe Leishmania with, to see how specific the attachment is in a model of permissive sandfly attachment. And so this is a kind of a representation of an atomic force microscope. And in essence, it's a very small cantilever with a tip, which you can functionalize with whatever you want, whether it be antibodies or protein. But in our case, we developed some quite unique chemistry to adhere these GalNAc mimics. And the tip is nanometers in width, and it taps over the surface of whatever you put in as a sample, in this case, promastigotes. And as it taps, any adhesion events will bend that cantilever and will be registered by the sensitive laser and photodetector. And you can quantify the amount, not only get a picture of where adhesion's happening over a promastigote, but the individual strength of those individual adhesion events. And so you can quite quickly build up a very powerful set of data just by analyzing several promastigotes effectively. And that's shown here. So what we were able to do was comparing metacyclic promastigotes versus nectamonad promastigotes. This is Leishmania mexicana. We can see that the adhesive force for GalNAc, which is representative of this mucin on permissive sandfly guts, there was much higher adhesion for the nectamonad promastigotes in the blue. And down here on the left-hand side, you can see some individual images of what those sort of adhesion maps look like for individual parasites. And we found that the distribution of adhesion events was mostly heterogeneous, so quite dispersed and random over the majority of sandfly promastigotes. But there was a significant proportion, around about 40%, of either nectamonad or metacyclic promastigotes, which had a stripe of strong adhesion. So the darker the color, the stronger the adhesion. They had a stripe of strong adhesion down the central axis of the cell body, which we think is potentially interesting, but also biologically relevant. We could maybe discuss that later. But nevertheless, what we were able to do was show that not only was the overall strength of the adhesion much higher for nectamonad promastigotes compared to metacyclic promastigotes, so stage specific, but it was also dependent on having LPG on the surface of those promastigotes as well, as you can see in the bar graph at the bottom right. And so we were able to conclude that unlike restrictive sandfly species and Leishmania combinations, which have a very specific protein receptor for a sugar on the LPG, here we have a new kind of mode of binding, if you like, which is between sugars. So it's attraction between sugars rather than a protein which has the capacity to bind a sugar. And so this is advancing our knowledge on the vectoral capacity of the permissive sandfly species out there. And another project I'd like to highlight is the one I've recently concluded with collaborators in the Natural History Museum in London and Martin Hall. And that's the use of a micro CT imaging facility to image infected sandflies and to get a handle on exactly the relationship of where the infection is, how big the infection is, where, how it deforms and manipulates the sandfly midgut in situ without dissecting or processing the fly beyond just preserving it in ethanol. And this involves a very expensive specialist bit of kit, the micro CT, which bombards a sample with X-rays as it's being rotated. And images are collected, 2D images, and then compiled by very powerful computers into the 3D models that we saw in the previous slide. And so here we have a sandfly that has gone from a 2D image to a 3D reconstruction on the far right, a sandfly which has no blood meal. And as you can see, the midgut, which has been highlighted in this stronger color, is very kind of squashed within the sandfly. And so we devised an experiment, quite simply, to compare uninfected flies versus infected flies, and those infected flies which were then fed a second time on an anesthetized mouse to gain a second blood meal, and to see the influence of a second blood meal on the anatomy of the sandfly and the anatomy of the infection. And these are representative images. You can appreciate that we scanned these individual sandflies for about 6 hours. Sometimes we're unsuccessful because the sample slips and you lose your imaging. So we, unfortunately, were only able to process a limited number of flies per group. But nevertheless, we did get some very striking images. And so we have control flies, day 7 and day 9 post blood feed, showing you that the midgut is squished within the sandfly and sort of compressed by the crop and the eggs in the anterior midgut, and is a very sort of thin tube during that runs through the thorax. However, in an infected fly, in a primary infected fly, in this bottom left-hand image, you can see that the midgut is expanded during infection. And we can even depict the gel within these sandflies in the stronger color inside each midgut. And you can see that it stretches from the stomatal valve right through into the abdominal midgut. And that's at day 9 post-infection. And on the far right, we have a primary infection at day 12 post-infection. And you can see that similar amounts of gel are present in those flies compared to the day 9 flies. However, if you give a fly at day 5 post-infection a second blood meal on a mouse and track them for until day 9, you can see that the gel and the gut is grossly distended. Is much bigger in these flies, which for the first time gives us a handle on the kind of volumes that we're dealing with in that scenario. And if we go in much higher magnification into one of these flies, we can see the gel in the middle of the anterior midgut here, which is really blocking and pushing open the stomatal valve. And not only doing that, but also gaining access and pushing itself into the pharynx of this particular infected sandfly, and also into the crop of that infected sandfly as well. And so this is a fly which acquired a second blood meal, and the gel really expands beyond the midgut in those infected sandflies. And this just summates the samples that we collected and we analyzed, and we can see that the presence of infection in green is much more anterior in those flies which have had a second blood meal, the T9 infected plus group of flies, compared to the others. And, you know, I mean, once you've collected these images, you can kind of play around with them to your heart's content, and you can really explore different aspects of the infection in much greater detail in situ. And one of them is that we tried to look at was how does the infection, and particularly the second blood meal infection, influence the volumes inside that midgut, and also the aperture of the stomatal valve. And what we found was that infection, whether it was a primary infection or a second blood meal infection, can grossly distend the midgut. But the stomatal valve opening, although it is much wider in infected sandflies, a second blood meal doesn't force it even wider. So you have a limit to how much it can open in terms of diameter. And this graph just shows you some of the analysis that we did on our limited number of samples that we collected. And by comparing lots of information that we collected on the volumes of the midgut and of the gel and the diameters of those stimuli valve, and subjected them to principal components analysis, we can see that the control uninfected group separates nicely from all the infected sandflies beneath it. And the just to draw your attention, those flies which received a second blood meal also are grouped quite distinctly away from the other infected groups. And so, just to summate this, oops, sorry. Yeah, the micro CT, we believe, is a powerful tool for providing a spatial temporal information on the progress of the infection in vectors, not just sandflies, but a wide variety of vectors. It can also provide a non-invasive and a non-destructive means, a virtual means of performing histology on any organ in that system. And there is a great potential to begin to answer a variety of questions, biological questions for parasite vector interactions. Unfortunately, we can't go down to the individual resolution of parasites using this particular machine, but there is prospect of doing that with other machines and other models. And nevertheless, what you generate is very impactful in terms of images and videos that can be used for all sorts of educational purposes. And so to just summarize some of the key things that hopefully I've introduced you to and touched upon, I think it's fair to say that Leishmania are experts at adapting sandflies for a very efficient transmission. Within that sort of study, we developed new methods, and not least the ability to determine the composition of the infectious dose from individual sandflies. And that revealed that the majority of bites delivered high quality transmissions of high proportions of metacyclics. And those sandflies, although I haven't shown you this data, can retain those high quality doses for up to 10 consecutive bites. So not only analyze one transmission event, but multiple transmission events from a single infected fly. And those high dose transmissions are more likely to contain non-infectious promastigote forms nevertheless. And this is via regurgitation, which is essentially set up by the accumulation of Promastigote Secretory Gel. And the Promastigote Secretory Gel also has the effect of retaining non-infectious promastigotes inside the gut, thereby making the infectious forms more available for transmission. And so we propose that the gel acts as a kind of molecular sieve, if you like, to allow infectious forms to be transmitted in relatively high doses. And so I've mentioned this, Leishmania adapt the sandflies for efficient transmissions. And this is in the face of a number of barriers that the sandfly presents to the Leishmania. These are nevertheless opportunities for control in the future, but not to interfere with vectoral competency. And that may include the amastigote transformation process and how it survives in the blood meal, how it escapes from the peritrophic matrix and then attaches to the midgut, and then how the process of metacyclogenesis happens and the gel formation, and hopefully we can exploit some of these and the other components of natural transmission as a transmission blocking vaccine. But in order to do all of that, any of that, we really must spend closer detail and time looking at these interactions in much greater detail. And so finally, are the acknowledgements, and there are many. And I've only picked out a few here which are relevant to some of the recent work that I've done. As Rod said, we go way back, and you know, we've had many conversations and collaborative projects between us. But within LSHTM, the UK, I need to particularly thank Dr. Emily Giraud, who's now at the Institut Pasteur, for her fantastic work with the Metacyclic qPCR. And more recently, collaboration with John Rains and Yushen Sao with the surface plasmon resonance. And more recently with Dr. Leith Jacob, who's a mathematical modeler. And currently my PhD student, Ms. Eve Doran, is really focusing in on the early events of attachment in a more quantitative way. We have ongoing collaborations with Dr. Pagin Walrad and Lawrence Wilson in York, and I cannot not mention the influence that Alvaro's had on me in Liverpool. Yeah, another friend who has been very supportive, and I hope to work closely with him more in the future. And then we have the Natural History Museum, which was the micro CT project in Newcastle, an excellent biophysicist, Mark Gagan, who has walked us through the atomic force microscopy, which has revealed some very striking things about attachment. And so I must mention my financial support, which has been mostly through the Biotechnology and Biological Sciences Research Council, the BBSRC from the UK, which has also supported a network which has funded smaller projects on black flies, sandflies, and midges, and also the Bill and Melinda Gates Foundation and the Wellcome Trust. So, and finally, I thank you for your persistence and attention, and I'll take any questions.
Well, thank you so much for the amazing talk, Matt. Can you hear me? It's fine?
Yeah.
Okay. So thank you so much for sharing this beautiful data. Congrats on its amazing work. There is a compliment here in Portuguese in the chat. Thank you for the talk. And I would like to make a couple questions. I know it's quite late in the UK, so we will try to respect that.
That's fine.
I would just, yeah. Well, it's
Sandflies and Leishmania, it's okay to talk late.
okay. Well, I will make one question and see if people gets excited to do more. I was wondering about this effect of the nectomonads in the transmission after the bite. You know, that was quite surprising. I was wondering if nectomonads could be a source for transmission blocking vaccines instead of the usual thinking. What do you think about that?
Yeah, that's a good question. Certainly, LPG has been way back when explored as a vaccine. I can't remember if it had much efficacy, but that's certainly based on nectomonad preparations. But you're right. I mean, the presence of these non-infectious promastigotes may have immune consequences that we could exploit. That we could potentially use that as a transmission blocking vaccine.
I don't know if you explored the immunology of this effect, but I was wondering if this is specific or it's a general effect that you could follow, that you could see in other infections or other pathogens. Is this specific or?
We don't know. We did some limited immunology for that paper, following up on those infectious bites, and or those infections with needle, and also the infectious bites, when we grouped them into the different numbers of parasites that they were, the different qualities of parasites they were transmitting, and we saw that there were some cytokines in the mixed population which would benefit the migration of neutrophils. But also we had early pro-inflammatory cytokines such as IL-1 beta, which can set up a very strong protective pro-inflammatory response against the parasites. But conversely, we also saw that there was high amounts expression of insulin growth factor 1 in the enriched metacyclic bites, and that is one of the main pathways to alternatively activate macrophages. So you've got this kind of it seems as though the mixed population is just activating the pro-inflammatory response really strongly, and that's to the detriment of the parasites. But nevertheless, there are still parasites there for transmission. But those sandflies which transmit high quality doses, you have less pro-inflammatory mediators and a more of a skewing of the macrophages towards an alternative state where they can host Leishmania.
Okay. Well, there are questions in Clarissa. Maya Monteiro wants to do a couple questions, and then Reggie.
Sure.
Please, Clarissa, step up.
Yes. So just to situate you, I used to work with insects, but now I work with inflammation in mammalian. So then I was thinking, because in macrophages, the Leishmania, they depend on and also in the blood, they use the lipids from lipid droplets, and when passing the blood, they also take lipids from lipoproteins. So I was thinking on about the maturation of the parasite in the gut, and how this parallels with the blood digestion. What are the requirements of the parasite in terms of glucose and lipids, and how it happens with the competition for the food with the fly, and how and there's more things, but I think this is already something, if you can comment on that. I don't know anything about the Leishmania inside the gut.
Yeah, it's not my specialization, but I do know that there will be competition for nutrients and resources between the parasites and the sandflies. the parasites can achieve very high numbers in sandflies, certainly by experimental infection. And certainly Rod's early work showed that digestion can be dramatically affected by the presence of infection. And so that simply indicates there is a kind of benefit for manipulating digestion for the parasite. There will be a demand certainly for lipids by the parasite, especially when they have to produce these large cell forms such as the nectarmonad promastigotes. So there is likely to be a demand for cholesterol and other lipids like that to perform these dramatic cell changes, and also division within the population as a whole. But it's something that I'm not an expert on, but I would bet my bottom dollar that there's going to be competition for resources.
It could be that this gel that is secreted and it's enhances the capacity of the infection.
Yeah,
could it be that it's also a source of nutrients for the parasite?
It's entirely possible. I think that's a good idea. And you know, I've never considered it as a kind of energy store, if you like, locking away energy or carbohydrates away from the sandfly, and maybe as a potential reservoir for the parasites to sustain the parasites. So that really is an interesting concept. I've never considered. And also you've got to appreciate that, again, from Rod's early work and his work focus has the presence of these commensal bacteria, which could also use the gel as a potential resource, energy resource, or nutrient resource. So, you know, there's another level of trophic interaction there that could potentially be happening. What is particularly interesting from the work of Debbie Smith's group, Johannes Dell, they found that the SHERP and HASP mutants, they make metacyclics in vitro, the certain mutants of these parasites can make metacyclics in vitro, but they can't inside sandflies. And they don't make a gel inside those sandflies as well. So the gel and metacyclogenesis go hand in hand. But what other kind of interactions are missing if you take away a gel, which may impact on the metacyclogenesis as well? You know, so there are other kind of interactions to consider here.
And so it's interesting. And also this mixed infections, does the different parasites activate macrophage the same time? Because I think the work that I've seen, we use only one type. I never thought always in the lab you put together, we don't have the natural infection, we just do in vitro experiments with macrophages and the parasite or dendritic cells or whatever. And so could it be that the mixed infections can confuse the immune system, like activate something and paralyze the macrophage?
I reckon that could entirely be possible, yeah. And they may have different PAMPs on their surface, which may engage with different toll-like receptors, perhaps. So you're right, there may be a kind of confusion.
Disguise or something.
Yeah, it could be, they could be part of immune evasion, and they're just there to kind of act as an antigenic smokescreen for allowing the metacyclics to then infect successfully. Or they may, as you say, confuse amastigotes. Macrophages.
Yeah.
Macrophages are quite pleiotropic. They're not, you can polarize them, but you can, under certain circumstances, kind of flip-flop them between states. So, yeah, there may be advantage to that. But what we do know is that the gel, if you combine the gel with parasites, even in highly those macrophages which are polarized to kill Leishmania by adding, say, interferon gamma and TNF alpha. If you add gel with your parasites during those in vitro infections, you can get a small but significant proportion of parasites still persist in those very highly inflammatory macrophages, which may be very relevant to the initial establishment of the infection in a host where you've potentially got a kind of a highly pro-inflammatory environment, which would kill the majority of parasites, but this gel allows them to latch on, to have a foothold in the host during that very dynamic and very harsh, immunologically harsh phase in the skin.
Interesting. Yeah. Yeah, and there's another curiosity. Is there any species of Leishmania that enters the fly, like to pass the gut barrier to really infect the fly?
So do you mean penetrate the gut? Yeah. No, that's not considered a natural part of the Leishmania life cycle. I know Rod has shown me pictures of some heavy infections where there is potential of that happening. And I've certainly in heavy infections looked at the hemocoelic fluid from those infected flies, and you can see a few parasites, but they look very bad. They look very apoptotic and not in good shape at all. So they may penetrate the sandfly midgut into the hemocoel, but I don't think that's a viable strategy for them.
But the fly fights the parasite
presumably
strongly?
Yeah, presumably. I don't know how. I mean, these sandflies are not unlike other hematophagous insects. They have hemocytes, so they have the potential, but I don't know exactly how. Leishmania.
So the fly is not suffering much of the infection.
Yes, that's actually an unappreciated fact. Actually, heavily infected sandflies have reduced longevity. Certainly, with the fly combinations that we have worked with in the lab, we've seen a contracted lifespan in heavily infected sandflies. But that's only present if you stress those sandflies. If you just keep them in a cage, they will live just as long as your controls. But if you stress them, so you subject them to cold temperatures, or you force them to fly, or you starve them of sugars, then they will die more quickly than the equivalent stressed flies that were uninfected.
Possibly because of the nutrition, they are missing. Yeah.
Possibly.
Of course, you don't know the Leishmania population in the gut in wild caught flies, so you know what's going on in the wild could be completely.
Rod's right, and there's only a few studies that have analyzed and quantified infections from wild caught flies, and they found really heavily infected sandflies on the very few occasions they've managed to quantify them. They may have had a second or a third blood meal, which may have augmented their infection, who knows. But I was curious to see that, you know, the very low infection levels in sandflies can produce quite or at least transmit high quality doses. So, and that may be more indicative of what's actually out there. I don't know. Certainly for primary infections.
Mhm.
I think Reggie has a question.
Hi, Reggie.
Reggie?
Hi, Matt. Congratulations.
Oh, thank you.
Excellent work you done. I have one question, quick question, and then a comment, please. First, my question is: do you have any experience with another Leishmania type infecting sandfly in this case?
So with Leishmania-sand?
Yeah, with Longipalpus, yes.
Yeah. So we've done work with Infantum, Shigazi, sorry.
Yeah.
So we've done model infections with Infantum,
Right.
and we've been more recently working with Donovani, in fact. Donovani and Leishmania major. And Donovani is very quick, very quick inside Leishmania-sand. It's actually more like Leishmania mexicana. Leishmania infantum is surprisingly slow during its infection.
Yeah, I'm asking this, Matt, because I never saw one. Well, I was working in the field and I was dissecting sandflies in a very high infection locality, you know, and I was dissecting Lutzomia cruzi. That's the Longipalp group, anyway.
Yeah.
And I never saw, for the first time, I saw a plug in a natural infection.
Yeah.
Very, very heavily with the PSG, you know.
Great.
Yes, I was really amazed, and I couldn't forget your work in the PO and everything, you know. And I was in the field. I was really unlucky because I couldn't take a photograph of it.
Oh.
But eventually, we isolated the parasite in a hamster and was infantum.
Oh, great.
So,
Okay.
so it was the first time I saw it, and I was really amazed with your work and all this. PSG is amazing because you know people don't believe we see there the number of promastigotes swimming around and fantastic.
Yeah,
You know?
I mean, we've done some second blood meal infections with our infantum, and you really get massive infections and distortion of the gut and the gel.
Thank you very much, Matt.
No, thank you, Reggie. It's great to speak to you.
Okay.
Taina Cavalcanti wants to make a quick question. Taina, please.
Hi, can you hear me?
Yep.
Hello, nice to meet you. I just have a quick question. I was actually curious to know why Haptomonad promastigotes are your favorite stage.
They're just very distinctive and they're obvious. I like obvious things. They're the most. No, they have that important job of getting from the blood meal up to the anterior midgut, and that's their sole purpose. And without them, you wouldn't have transmission. So I think they're an important cell stage, and they're just really nice to see, because if you're kind of just trying to decide, oh, is that a metacyclic versus a late stage Haptomonad promastigote, and then you've got a big Haptomonad next to it, you know what you're talking about with a Haptomonad.
Thank you. I was just asking because my favorite stage is Metacyclic, so I was just curious to know. Thank you so much.
Thanks. Someone wants to make another question, or? Well, I will do just one more. Sorry, it's. Do you think this low parasite load in high infective bites, this is a parasite strategy or a consequence of the sandfly response? What do you think about that?
Sorry, could you say that one again?
Because you have these bites with that are very infective, but with very low parasite load.
Yes, yes.
So this low, so you have two populations of flies, and one of them is the two populations of bites, sorry, and one of them is so it looks like a strategy. So having few parasites. So do you think this is a strategy of the parasite, maybe related to population detection, to the amount of parasites, or do you think this is just a consequence of the sandfly response to infection?
Well, it's very tempting to call it a strategy. I mean, all these flies were infected with the same dose, prospectively. So yeah, you could. It's very tempting to say it's a strategy, and it's certainly what may be present in the wild, because it's. I do concur that sandflies are likely to pick up much lower doses than we use for experimental infection. But I think, and we've got some data, but we think that the bacterial composition of sandflies can really influence the infection in those sandflies. And so, as I mentioned earlier, there's a whole host of other trophic interactions going on. So, do you know, it's so exciting to call it a strategy, and I can see the benefit that you minimize your impact on the vector. The vector's healthy. The vector can feed efficiently. You don't impact on the fecundity, perhaps, of those vectors, and yet you are still able to be transmitted, and it's so attractive. Yeah, I can't say either way, but yeah, the biologist in me is wanting it to be a strategy.
Okay, Rod has a question. Rod?
Sorry to prolong everything. I just wondered if Matt could sort of gaze in his crystal ball sort of 10 years forward and sort of think about what would be the situation with sort of Leishmania-sand flies and research in 10 years' time, and sort of we'd be still working in the lab, you'd be still working in the field. What would be going on? What would you be doing?
Well, I hope I would still be in the lab. Yeah, I think there's this, you know, I'm just a simple biologist. I like things to be simple, and what I do is essentially simple. I infect sandflies and I look at the infection. But there are so many other ways of analyzing this interaction that we just haven't considered yet. And I've scratched on a few, you know, I've touched upon a few, atomic force microscopy, the micro CT. I mean, the atomic force microscopy, take that for example, you get so much data out of so few parasites that you could then pass on to a modeler perhaps, to then can really drill into and come up with hypotheses that you never dreamed of. So I really think there is reaching out to other fields. It will be very fruitful for Leishmania-sandfly interaction research. And I really hope that we can get to a point where we have good candidates for transmission blocking vaccines or transmission blockade to interfere with transmission. I really hope we can, whether it's new vaccines or it's repurposing old drugs which interact, prevent key stages of the Leishmania-sandfly interaction or transmission itself. So I'm hoping that we can provide that information to advance that in particular. But also in the field, definitely in the field, I mean, there's massive demand and gaps in our epidemiological knowledge regarding transmission. You know, we're still going out there and collecting thousands of insects to process, admittedly better because it's PCR, but certainly it'd be really good to link the biology, what we know about transmission, with the epidemiological data we can collect, you know, as part of longitudinal studies to better model it.
Great. Thanks.
Well, somebody wants to make a last one, or I think we are. Beto, do you want to?
I just wanted to make a comment, in fact, because you have a very interesting presentation. It was really fantastic. You use a very simple strategy to answer very good biological questions. I think that was really amazing.
Thank you.
So thank you for that. But so one thing that was very interesting was something related to what Fernando was mentioned before, and also Clarissa, about the immune response to the mixed parasites. So is there any specific molecule that is expressed by Nectomonad that could be used as a candidate for a vaccine? So is there any known molecule, for example, exosome-derived molecule that is secreted by the Nectomonad?
I'd have to, I mean, there are some recent studies that looked at using RNA-seq to really drill into these different parasite populations, not least in culture, but also from sandflies themselves. So I think there probably are a selection, but I think that could be the evolution of this work and other people's work, where we really use those markers to reanalyze the interaction, but also look at transmission and infection as well. On the top of my head, I'm sorry, I don't know of a Nectomonad specific one. Although I know, although I do know that, for example, people looking at RNA transcription factors, that seems promising to separate different promastigote stages, as well as, you know, anything that's kind of surface expressed.
Yeah, it's really interesting. I haven't seen anything before. So like a co-infection with two different stages of Leishmania. It was really interesting. So it sheds light on a new strategy to searching for a molecule that is produced by Nectomonad that could be used as a potential candidate for a vaccine. So yeah. Interesting, very interesting.
Yeah, and they're pretty easy to get hold of in culture as well, particularly if you go from lesion amastigote material straight into culture. If you time your cultures correctly, and you've got the additional kind of safety net of doing phycol purification, you can get very high populations of Nectomonads.
So a very simple strategy, just wondering, so if you injected that Nectomonad, for example, in mice, and you try to infect with Metacyclic ones, what could happen?
Good question. So concomitant immunity is what was essentially found using Leishmania, and that's where one Leishmania can protect against reinfection from another Leishmania, and that can cross species. So we know that I think it's infant can protect against Leishmania major infection, as well as Leishmania major or tropica protecting against the same parasite. So yeah, I mean that's a great question, and I know that some people are modifying Leishmania to essentially infect but then die within the host, or are non-replicative in the host, and that they can establish this phenomenon of concomitant immunity and protect leishmanization essentially, and protect the host.
Thank you.
Oh, well, I think we should close the session because it's getting late. It's getting late even in Brazil. So
Yeah, so
I want to thank you, Matt, thank Rod, thank Matt for the amazing session. I'm very grateful and hope to see you soon.
Yeah, I hope so too. And thank you everybody for listening.
Thanks, Matt.
Bye, Matt.
Okay.
Bye-bye, Juan.
Bye-bye.
nice to see everybody.
Okay, thank you.
Thank you.
Bye bye.
Everyone.
Take
Thank you,
care.
everyone.
Bye.
Take care.